Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology, Unit 1 for October 2024. Let's begin with the first question. In question 1, amoeba are single-celled organisms that live in water. They are able to change their shape. The diagram shows two different shapes that one amoeba can make, so we see shape 1 and shape 2. The membrane of shape 2 has projections, while that of shape 1 doesn't. In part A, an amoeba changes shape by pushing cytoplasm against the cell membrane, forming long, thin projections. They ask, state the property of the cell membrane that enables the amoeba to change shape. Because the cell membrane is extended due to its fluid nature, the answer is fluidity. In part B, gas exchange in an amoeba takes place across its cell membrane. The amoeba can be roughly spherical, as shown in shape 1. They ask, which is the surface area of the amoeba in this shape? They have given us the formula for surface area of a sphere. From shape 1 above, the diameter is 300 micrometers. The radius is therefore 150 micrometers. So, area equals 4 times pi times 150 squared. The answer is 90,000 pi. The answer is therefore A. Next, they say, the mass of the amoeba stays the same when it changes from shape 1 to shape 2. They ask, which row of the table shows what happens to the surface area and volume of this amoeba when it changes to shape 2? The surface area increases due to the amoeba spreading around. The volume stays the same. Next, they say, explain why an amoeba can rely on diffusion to supply the oxygen it needs. Relying on diffusion can be because of the surface area to volume ratio, the concentration gradient, and the diffusion distance. So I said it has low energy requirements. The oxygen concentration of the surroundings is higher than that of the cytoplasm, providing a steep gradient. Also, there is a short diffusion distance from the surroundings to the cytoplasm. In question 2, during the development of a human, the structure of hemoglobin changes. In the developing fetus, the main oxygen-carrying molecule is fetal hemoglobin. Towards the end of fetal development, fetal hemoglobin is replaced by adult forms of hemoglobin. In part A, they say, blood may contain 5.3 million red blood cells in 1 millimeter cubed of blood. One red blood cell can contain 270 million hemoglobin molecules. They ask us to calculate the number of hemoglobin molecules in 1 millimeter cubed of blood and express the answer in standard form. First, I multiplied 5.3 times 10 to the power of 6 by 2.7 times 10 to the power of 8. That gave me 1.43. 1 times 10 to the power of 15 hemoglobin molecules in 1 millimeter cubed of blood. Next, they say, which of the following statements about the structure of hemoglobin are correct? The statements given are, it is a fibrous protein, it has a quaternary structure, it contains a non-protein heme group. So the question is testing if we know what type of protein hemoglobin is and what structures it has. Hemoglobin is not fibrous, but it's globular, but it does have a quaternary structure and it does have a heme group. Therefore, the correct answer is D. Statements 2 and 3 are correct. Next, they say, the graph shows the changes for 3 types of hemoglobin subunits present during each stage of development. The graph tracks the percentage of 3 types of hemoglobin subunits, alpha, beta, and gamma, across different stages of development, embryonic development, fetal development, and after birth. They ask, describe the changes in these subunits during each stage. So I said, during embryonic development, the alpha, beta, and gamma subunits all increase. During fetal development, the alpha subunit remains fairly constant, the beta subunit starts to increase, and the gamma subunit starts to decrease. After birth, in the baby, the alpha subunit remains constant, the beta subunit continues to increase and then levels off, and the gamma subunit continues to decrease and then levels off. Next, they say, the oxygen affinity of fetal hemoglobin is different from adult hemoglobin. Explain why this difference ensures the fetus obtains enough oxygen during development. So this part is asking us to think about how fetal hemoglobin helps the fetus get enough oxygen during development. So I said fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. This means that At the same partial pressure of oxygen, the oxygen will leave the mother's adult hemoglobin and bind to the fetal hemoglobin. This way, the fetus is able to absorb enough oxygen for its growing tissues at low partial pressures of oxygen. In question 3, they say, Charcot-Marie tooth, CMT, is an inherited disease that results in the gradual loss of function and sensation in the hands, arms, legs, and feet. Over 100 different gene mutations have been identified that cause CMT. The pedigree diagram shows the inheritance of one form of CMT in part of a family. The allele for CMT can be represented by the letter C in this recessive pattern of inheritance. Now, in this question, they are setting the background. They explain that CMT is recessive, meaning 2 copies of the faulty allele are needed to develop the disease. They also show a family tree that we need to use to answer the next parts. In the first part, they say, which row of the table shows the genotype and phenotype for individual 1? So here they want us to look at individual 1 in the pedigree chart and figure out both their genetic makeup, genotype, and whether they show symptoms, phenotype. So I said, looking at the diagram, individual 1 is unaffected. They don't show signs of CMT. Since the condition is recessive, an unaffected person could either be CC, homozygous normal, or CC, a carrier. Based on the diagram and options given, individual 1 is most likely CC, a carrier. The correct answer is B. Next, they say, which allele combinations could individual 3 have? Now they're asking about individual 3's possible genotypes. Since both parents could be carriers, we have to think carefully about what alleles individual 3 might have inherited. So I said, individual 3 is unaffected, so they could either be CC, 2 normal alleles, or CC, 1 normal and 1 faulty allele, making them a carrier. Therefore, the answer is D. Next, what is the probability that a 4th child would be a female with CMT? This time, it's a 2-step probability question. First, the chance of getting CMT, then the chance of being female. So I said, the chance of inheriting CMT is 1 in 4 because it's recessive and both parents are carriers. The chance of being female is 1 in 2. Multiplying those together, 1/4 times 1/2 equals 1 in 8. The answer is C. Next, they say, other forms of CMT show a dominant pattern of inheritance. In the USA, 5% of people with CMT have one of these dominant forms of the disease. In the USA, there are a total of 126,000 people with all forms of CMT. Calculate the number of people in the USA who have one of these dominant forms of CMT. This part is just a straightforward percentage calculation. I multiplied 126,000 by 0.05. That gave me 6,300 people with a dominant form of CMT. Next, they say, an individual with a dominant form of CMT will have the allele combination TT homozygous dominant or TT heterozygous. Draw a genetic diagram to show the possible allele combinations of the children, where one parent does not have CMT and the other parent has the genotype TT. Here they're asking for a Punnett square, showing a cross between a normal parent and a carrier parent. So I said, the unaffected parent is homozygous recessive and the carrier parent is heterozygous. Therefore, half the children would have CMT and half would not. They say, another form of CMT can be inherited as a sex-linked trait on the X chromosome. Explain why a male with CMT cannot pass the disease to his sons. This part focuses on how sex-linked traits are inherited. So I said, a male passes his Y chromosome to his sons, not his X chromosome. Since the CMT gene is carried on the X chromosome, he cannot pass it to his sons. Only his daughters would inherit his X chromosome, not his sons. In question 4, they say, the circulatory system and the cardiac cycle ensure that all the cells of the body are supplied with their requirements. Here they are reminding us that the body needs a constant controlled flow of blood to deliver oxygen and nutrients and to remove waste. They say, the table shows the diameter of the lumens of an aorta and a capillary and the thickness of their walls. This part is about comparing the size and wall thickness between the aorta and the capillary, two very different types of blood vessels. In the first part, they say, how many times greater is the diameter of the lumen of the aorta than the diameter of the lumen of the capillary. They want us to calculate how much bigger the aorta's lumen is compared to the capillaries. But we have to be careful with units because the aorta is measured in centimeters and the capillary in micrometers. I converted the units first. 2.5 centimeters equals 25,000 micrometers. 0.14 micrometers stays as 0.14 micrometers. Then I divided. 25,000 divided by 0.14 equals about 179,000 times greater. So the correct answer is D, 179,000. Next, they say, explain why the wall of the aorta is thicker than the wall of a capillary. Here they want us to link the structure to the function of these vessels. So I said, the wall of the aorta is thicker because it must withstand the very high pressure of blood being pumped directly from the heart. It contains elastic fibers, smooth muscle, and collagen to allow it to stretch and recoil, helping to maintain blood pressure between heartbeats. In contrast, the capillary has a very thin wall, only 1 cell thick, because its main job is efficient diffusion of substances like oxygen, carbon dioxide, and nutrients between blood and body cells. Thin walls reduce the diffusion distance. In part B, they say, the table shows the durations of ventricular diastole and ventricular systole at 2 different heart rates. They ask, determine the relationship between heart rate and the duration of ventricular diastole and ventricular systole. This part is testing whether we can see the pattern between heart rate and how long each phase of the heartbeat lasts. So I said, as the heart rate increases, the duration of both ventricular diastole and systole decreases. However, diastole decreases much more sharply than systole. This shows that at very high heart rates, the heart reduces its resting time, diastole, much more than its contraction time, systole. The graph shows the rate of flow of blood through the aorta of a dog with increasing distance from the heart. Based on the graph, there are sharp peaks in forward blood flow near the heart. Also, here is gradual lowering of peak forward flow further away from the heart. Small amounts of backward flow appearing further from the heart. They ask, explain the changes in rate and direction of flow of blood through this aorta. Use the information in the graph to support your answer. Here they want us to interpret the graph and explain why forward flow decreases and backward flow increases as distance from the heart increases. So I said, near the heart, there are high peaks of forward blood flow because the blood is being forcefully pumped by the heart during systole. As the distance from the heart increases, the peak forward flow decreases because of loss of pressure, resistance from vessel walls, and branching of the aorta into smaller arteries. Backward flow becomes more noticeable further away from the heart because during diastole, there is no new force pushing the blood forward, and the elasticity of the arteries alone isn't enough to fully prevent slight backflow. In question 5, they say Warfarin is used as an anticoagulant to treat some patients. Patients respond differently to this drug. This makes it difficult for doctors to select a safe but effective dose of Warfarin to give to each patient. Factors that affect the response of a patient to Warfarin include age, body mass, liver function, and genetic makeup. Here they are explaining that Warfarin dosing is complicated because people's bodies and genes affect how they react to the medicine. Some people might process Warfarin faster, slower, or need a different amount for it to work safely. They say, explain why a safe but effective dose of Warfarin needs to be given to a patient. This part is asking why it's so important to carefully balance the dose, not too much and not too little. So I said, if the dose of Warfarin is too high, it can cause excessive bleeding because the blood cannot clot properly. If the dose is too low, it might not prevent blood clots, which can cause serious problems like strokes. So a safe but effective dose is needed to prevent clotting without causing dangerous bleeding. Next, they say, there are 2 genes that code for proteins that affect warfarin and the blood clotting process. Gene M codes for an enzyme M that breaks down warfarin into inactive molecules, has many alleles, and some versions cause slower breakdown of warfarin. Gene V codes for enzyme V that converts vitamin K into its active form, triggering the blood clotting process. Some versions result in decreased production of mRNA for enzyme V. Here they are giving us information about how genetics, specifically two genes, M and V, can affect how a person responds to warfarin. Gene M affects how fast warfarin is broken down, and gene V affects how vitamin K is activated for clotting. Then, describe the role of mRNA in the production of enzymes. They are asking about the general biological role of mRNA, especially related to how proteins like enzymes are made. So I said, mRNA carries the genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm. At the ribosome, the sequence of bases in the mRNA is used to determine the sequence of amino acids, which are assembled to form a specific enzyme through translation. Pharmacogenomics analyzes the genetic profile of individuals. This analysis is used to determine the appropriate doses of a drug for individual patients. They ask, discuss the possible use of pharmacogenomics in selecting appropriate doses of warfarin for individual patients. Use the information in the question and your own knowledge of the blood clotting process and genetic screening to support your answer. This is a 6-mark essay style question. They want a developed answer using ideas about genetics, clotting, and why personalizing doses is important. So I said pharmacogenomics can help doctors choose a safer, more effective dose of warfarin for each patient by looking at their individual genetic profile. Some people have genetic variations that cause enzyme M to break down warfarin more slowly. If the breakdown is slower, warfarin stays longer in the blood, meaning a lower dose is needed to avoid serious bleeding. Other patients may have variations in the gene for enzyme V, which affects vitamin K activation. If less enzyme V is made, clotting will already be reduced, so an even smaller dose of warfarin would be needed. Pharmacogenomics also considers other factors that influence warfarin dosage, such as age, body mass, and liver function, which can all affect how the drug is processed. However, using genetic screening raises ethical and practical issues. Patients must give informed consent before testing, and there are concerns about the confidentiality of genetic information. Patients may also experience anxiety or stress after learning about their genetic risks. Despite these concerns, pharmacogenomics improves the safety and success of warfarin treatment by personalizing doses to match the patient's needs, helping to prevent dangerous bleeding or clotting complications. In question 6, they say the photograph shows some soybeans. Here they are introducing soybeans, which are seeds inside pods. The rest of the question is about how eating soybeans might affect cholesterol levels and heart health. From the first part, eating soybeans may reduce cholesterol and LDLs in the blood. They ask, explain why eating soybeans may reduce the risk of heart disease. This part is about linking cholesterol and LDLs to heart disease and explaining how soybeans help. So I said, eating soybeans can help to lower the amount of LDL cholesterol in the blood. LDL cholesterol can deposit fatty substances inside arteries, leading to atherosclerosis. By reducing LDL levels, soybeans lower the risk of plaque buildup, keeping arteries clearer and reducing the risk of heart disease. In part B, they say, the extent to which soybeans reduce cholesterol depends on the variety of soybeans. This reduction is influenced by two proteins found in soybeans, glycinin and beta-conglycinin. They ask, give 2 ways that the primary structure of two proteins could differ from each other. Here they are asking about the basic idea of primary protein structure, which is simply the sequence of amino acids. So I said, the two proteins could differ in the number of amino acids they have and the sequence or order of their amino acids. Next, they say the effect of soybean extracts on cholesterol production by liver cells growing in culture was investigated. Scientists produced fat-free soybean flour and digested it with enzymes mimicking digestion in the human body. They show a diagram describing the steps from mixing with saliva to heating the mixture. Here they are showing how they prepared the soybean extract by mimicking digestion realistically, using different enzymes and body temperatures. In part CI, they say, explain why the scientists used digested soybeans to make the extract for this investigation. They are asking why the soybeans needed to be digested first before testing. So I said, the scientists used digested soybeans because in the human body, soybeans are broken down by enzymes during digestion. Using digested soybeans more accurately mimics how soybean peptides would be produced and absorbed naturally in the human digestive system. Next, they say, suggest why the soybean flour was incubated with different enzymes in steps 2 and 3. This part is about using the right enzymes for each stage of digestion. So I said different enzymes are needed to mimic digestion in different parts of the body. Stomach enzymes break down proteins in acidic conditions, while pancreatic enzymes continue digestion in the intestines under alkaline conditions. Next, they say, explain why the soybean flour and enzymes were incubated at 37 degrees Celsius for 2 hours in steps 2 and 3. Now they are asking why they use these specific conditions. So I said, 37 degrees Celsius is the normal human body temperature, which allows the enzymes to work at their optimum rate during digestion. Incubating for 2 hours gives enough time for the enzymes to break down the soybean proteins properly, similar to how long food would stay in the stomach and intestines during real digestion. In part D, they say the soybean peptides at different concentrations were incubated with liver cells growing in culture. The enzyme HMGCR is involved in the synthesis of cholesterol. The effect of soybean peptides on the activity of this enzyme and the synthesis of cholesterol was measured. They show a graph. As the concentration of soybean peptides increases, inhibition of HMGCR activity increases. Meanwhile, synthesis of cholesterol decreases. They ask, explain why eating soybeans may reduce cholesterol levels in the blood. Use the information in the graph to support your answer. Here they want us to explain how soybean peptides affect HMGCR and how this reduces cholesterol using the graph as evidence. So I said, as the concentration of soybean peptides increases, the inhibition of HMGCR enzyme activity also increases. HMGCR is the enzyme responsible for cholesterol synthesis in the liver. When HMGCR activity is inhibited, it reduces the production of cholesterol inside the liver cells. The graph shows that as the inhibition goes up, the rate of cholesterol synthesis falls. Therefore, eating soybeans, which provides the soybean peptides, reduces the activity of HMGCR, leading to a decrease in cholesterol production and lowering the overall blood cholesterol levels. This ultimately reduces the risk of developing heart disease linked to high cholesterol. In question 7, they say, scientists investigated the numbers and types of mutations in different species of animals. Samples of tissue were collected from living animals of each species, and the types of mutations were determined. The types of mutation were classed as either substitution mutations or insertion and deletion mutations. They show a table with results. The table shows the number of animals sampled for each species and the range of mutation numbers for substitution and insertion or deletion types. Here they are studying genetic mutations across animals like naked mole rats, mice, dogs, and humans. They are comparing how many mutations happen and what types. Next, they say, describe how these types of mutation affect the DNA-based sequence of a gene. This part is asking how substitution, insertion, and deletion mutations change the sequence of bases in DNA. So I said, a substitution mutation replaces one base with a different base. An insertion mutation adds one or more extra bases into the sequence. A deletion mutation removes one or more bases from the sequence. All of these changes alter the DNA-based sequence of the gene. Next, they say, explain why these animals can survive with a greater number of substitution mutations than insertion and deletion mutations. This part is asking why substitution mutations are less harmful compared to insertion, deletion mutations. So I said, substitution mutations only change one base, and sometimes they are silent mutations, meaning they don't change the amino acid made. Even if the amino acid changes, the overall shape and function of the protein might still work normally. Insertion and deletion mutations are usually more harmful because they cause a frame shift, which changes every amino acid after the mutation, leading to a non-functional protein. Therefore, animals can survive better with more substitution mutations than with insertions or deletions. Next, they say, suggest which set of data is the most valid, give a reason for your answer, This part is asking which animal's data is the most reliable. So I said the mouse data is the most valid because the sample size is the largest. 10 mice were sampled, which means the results are more representative and more reliable. Moving to part B, they say the graphs show the number of mutations per cell during the lifespans of the 4 animals. They show scatter plots for naked mole rats, mice, dogs, and humans. Here they are giving real data on how mutations accumulate as animals age. Next, they say calculate the mean mutation rate for the mouse. Draw a line of best fit onto the graph to help you. This part is asking for a rough mean mutation rate based on the mouse graph. So I said, looking at the graph and estimating the best line of fit at 0 years, about 300 mutations, at 2 years, about 1800 mutations, change in number of mutations equals 1800 minus 300 equals 1500 mutations, change in time equals 2 years, mutation rate equals 1500 divided by 2 equals 750 mutations per cell per year. Then they say, describe 3 conclusions that can be made from these graphs. This part wants us to pull out patterns from the graphs, so I said, the number of mutations per cell increases with age in all species. Dogs and humans have higher numbers of mutations per cell compared to naked mole rats and mice. The rate of mutation accumulation is faster in mice compared to the other species because their lifespan is much shorter, but mutations accumulate quickly. Next, they say, suggest 2 reasons for the differences in the number of mutations per cell of these species of animal. This part wants ideas about why mutation numbers vary between species. So I said, differences in lifespan. Longer-lived animals have more time to accumulate to mutations. Differences in metabolic rate. Higher metabolic rates can produce more reactive oxygen species that cause more mutations. Differences in DNA repair mechanisms and different rates of cell division. In question 8, they say, the photograph shows 2 avocados. Here they are introducing avocados because they are rich in unsaturated fatty acids, and this leads into a question about comparing fats. In part A, they say, avocados are high in unsaturated fatty acids and low in saturated fatty acids. Compare and contrast the structure of an unsaturated fatty acid and the structure of a saturated fatty acid. This part is asking us about the chemical structure differences between saturated and unsaturated fatty acids. So I said, an unsaturated fatty acid contains at least 1 double bond between carbon atoms in the hydrocarbon chain. A saturated fatty acid has no double bonds. All the carbon atoms are fully saturated with hydrogen. The presence of double bonds in unsaturated fatty acids creates kinks in the chain, making them less straight compared to saturated fatty acids, which have straight chains. In part B, they say, the effect of eating avocados each week on the risk of coronary heart disease, CHD, was investigated. A group of men and women with no signs of cardiovascular disease, CVD, or stroke were selected. They were all between the ages of 30 and 75. The group ate avocados weekly to replace animal products in their diet whenever possible. They completed a questionnaire at the start and every 4 years for over 30 years. Here they are setting up a long-term investigation about diet and heart disease risk, focusing on avocado intake. They say there were 41,701 men and 68,786 women in the group, which is the ratio of men to women. This part is a simple ratio calculation. So I said, to calculate the ratio of men to women, 41,701 divided by 68,786 is approximately 0.606. This matches the ratio of 0.6 to 1, so the correct answer is A, 0.6 to 1. Then they say, describe the questions that need to be in the questionnaire so that appropriate information can be gathered for this investigation. Give reasons for choosing these questions. This is a 6-mark question asking for thoughtful ideas about designing a good questionnaire. So I said the questionnaire should include the following questions. How often do you eat avocados each week? This measures the frequency of avocado consumption, which is the key variable being investigated. How many portions of avocado do you eat per serving? This collects quantitative data about the amount of avocado eaten, helping to standardize intake across participants. Do you replace animal products with avocados in your meals? This checks if participants are actually substituting animal fats, not just adding avocado on top of their normal diet. What other fruits, vegetables, and fats do you regularly eat? This identifies other dietary factors that might influence heart health. Do you currently have, or have you ever had, cardiovascular disease, high blood pressure, or high cholesterol? This screens for pre-existing health conditions that could affect the results. How often do you exercise each week, and what type of exercise do you do? Because physical activity levels are strongly linked to heart health and need to be considered. Do you smoke, or have you ever smoked? Smoking is a major independent risk factor for heart disease, so it's essential to record. How much alcohol do you consume per week? Alcohol intake can affect heart health, so it's important to control for. What is your age and biological sex? Both age and sex impact heart disease risk. Older individuals and men typically have higher risks. Do you have a family history of cardiovascular disease? Genetics can predispose someone to heart problems regardless of diet. Do you take any medication related to cholesterol or blood pressure? Because medications could alter blood cholesterol levels, confounding the results. Next, they say, comment on the validity of the methods used in this investigation. They want us to critically evaluate the setup of the avocado heart disease study. So I said, the investigation has strengths because it uses a large sample size and a long duration, over 30 years, which increases reliability and allows long-term trends to be seen. However, self-reported questionnaires may introduce bias or inaccuracies because people might misreport their food intake. Also, other lifestyle factors like diet, exercise, smoking, and alcohol can confound the results, making it hard to prove that avocado intake alone affected heart disease risk. So this brings us to the end of the paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.